episode 2 of Cass Talks, Cass will share some results from a survey he did with members of the public in Singapore. We uncovered some myths and misconceptions. Introducing to you our top 4 misconceptions. Coming in at number 4, DNA bases in a gene cannot be changed or altered in our body. This is false. DNA bases in a gene can actually be altered in our body. Let's hear from the experts.
This statement says in our body, okay, they are being altered and changed by random mutations all the time. Everybody carries different mutations. I mean, if you spend too much time on Sentosa on the beach, you get lots of mutations in your skin cells, and eventually that won't be good.
Our experts have shown we can alter our DNA bases. Coming in at number 3, current genome editing techniques are able to edit or make changes to all genetic information in the cell. This is also false, and the experts will tell you more.
This statement kind of suggests that you can change or you can target any sequence and you can do all the changes in that place, which again is not possible. Even within all the regions that you can target, some regions will be very difficult to, make any changes, like with the homologous direct repair or whatever, because this is very inefficient. Depending on the cells that you're working on, some cells don't have the necessary protein components to do any homologous recombination. You may be able to get around that with the base editors, but again, even with the base editors, you're limited by your guide. You have to have the guide at the correct spot, and even within the guide sequence, it's only that those few bases within those 20 that the editor will efficiently target. So again, if you don't have a good guide at the spot that you're wanting to target, there's nothing you can do. So that's one of the limitations we have with our study currently, is that we have some candidates that we simply cannot target because there's no guide in that region that will be specific enough for us to use. So we can't do anything about it. There's obviously we try some other things, like if the base editor can't work, we try that if we can try it with homologous directed repair. But again, you need a guide that works. If you don't have a guide that works in the space, there's very little you can do with the current techniques. Obviously, as I said, more and more Cas proteins are being discovered, and some of them will open up new avenues. But as it stands, and I'm sure for quite a while still, this statement is false.
No, that's not true either, because even for genome sequencing, we haven't sequenced every part of the genome. There are parts of the genome that are very difficult to sequence. And to be perfectly honest, we don't even know how many real genes there are. I think we have a pretty good idea of the approximate number of protein coding genes, but we know that there are transcription units that make RNAs, which actually function as RNAs. They don't make proteins. So nature didn't intend these RNAs to function as proteins, and we have no idea. The RNA world is still opening up. So if we include the RNA genes, that would be a whole lot of genes. So I don't think it will ever be possible for us to edit or change every single gene.
Now that we have busted 2 myths, tune in to our next episode of Cass Talks for the top 2 misconceptions.